Our next unit, forensics and DNA. In 1980s and mid-1980s, DNA fingerprinting dramatically changed forensic science and the ability of law enforcement to link perpetrators with the crime scenes. In early 1990s, the U.S. military began collecting and storing the blood samples of soldiers. So they actually took the DNA from the soldiers. And this is pretty invasive because your DNA pretty much tells everything about you, from your family history to your medical history. All the conditions you have is contained in your DNA. In 1998, they began a national DNA index system, much like the fingerprint system we talked about before, that database, and it was renamed CODIS for Combined DNA Index System, I believe in 2000. By 2014, CODIS had actually 11.1 million offenders, their DNA profiles, and 1.9 million of DNA profiles of just people who were arrested. So in 2013, yes, if you were just arrested, they could take your DNA. If we're going to understand how DNA is used in forensics, we've got to go back and do some review on what is DNA. So here we take a look at the double helix. So these base pairs on the double helix are joining together the two strands of the DNA. If I look at your chromosomes, and I can do what's called a karyotype here, I can take your chromosomes and lay them next to each other according to chromosomes that are alike. And so those chromosomes that are alike, you'll recall from previous years, those are called homologous pairs of chromosomes. So those are important. The homologous pairs of chromosomes, they're the same in that the same genes are found on each of the chromosomes. So you have 23 pairs or 46 total chromosomes, one of which you got from mom, the other of which you got from dad. So here's another karyotype, and you'll notice in this karyotype, we have 3 copies of the 21st chromosome, 3 copies of this chromosome. Now I have that much more information, which gets turned into that much more product. This, by the way, is known as Down syndrome. This trisomy 21 is Down syndrome, having a 3rd copy of that 21st chromosome. So what represents the genes? To understand that, we've got to take a look at more details for this deoxyribonucleic acid. So here, once again, is DNA. We've broken it down to the simplest part. So this part here, this is known as the nucleotide. So a phosphate, a sugar, and a base. This is the simplest part of DNA. This is the monomer that makes up the bigger polymers. If I take one nucleotide, we see here again, we got the phosphate, the sugar, and the base. If I take this nucleotide and I combine it with another nucleotide, I start to build my bigger molecule, the polymer of DNA. So here's one nucleotide being joined to another nucleotide, and I'm now making my polymer we call deoxyribonucleic acid. Notice the negative oxygen here on each one of these phosphate groups. That'll be important in terms of how we move DNA through a gel. So here's the longer structure. Once again, I've got a base here, my sugar, and my phosphate. Repeat these all the way down. I've got these 1, 2, 3, 4 nucleotides joined together forming my DNA. Here are the 4 different kinds of bases. I have cytosine, adenine, guanine, and thymine. And so I have these nitrogen containing bases. They are the most important part of DNA. So they bind together to form my double helix. So these complementary sequences. So thymine is going to join together with the base adenine. I have a strand of DNA on this side and a strand of DNA on this side. So when we say that DNA is double-stranded, that means we have 2 strands of these polymers joined together. And they always join together in the same way. These bases, adenine will join together with thymine. Guanine joins together with cytosine. In case you ever want to remember that, memorize that, always think straight lines with straight lines, curved lines with curved lines. So A with T and G with C. Notice how these are attached together. We have hydrogen bonds attaching to my bases. Remember, those hydrogen bonds are a fairly weak bond. They're easy to break apart, and that's really important in terms of forensics as well. So here then is our double helix of DNA. Once again, 2 strands of DNA are joined together. So I've got a strand on top, strand on bottom, that join together at the bases. So we want to discuss the difference between genotype and phenotype as well here. So genotype, as it turns out, the genes are your sequence of bases. So I could have a C, T, C, or I could have an A. That sequence of bases is the genotype. The phenotype is what that gets turned into. So let's take a look at that. As we look at DNA, we know that is the information, and that gets turned into a message, and that message then gets turned into a product, which is the protein. So DNA is the information, mRNA is the message, and the protein is the actual product. So everything about you is written in the code of those bases. The sequence of bases is in fact a gene. If we look at a gene, then there are two kinds of things involved in DNA. Some things we call exons, other things we call introns. And we're going to take that gene then and turn that into a message. So here's our mRNA transcript. Exons, introns, exon 2, intron, exon. And then that actually gets modified, and what gets cut out then are the introns. So I've got this area in the DNA that doesn't really code for anything. They exist, and they're really, really important in terms of forensics, but they don't code for anything. The exons are what code for that protein we saw before. Let me show you a video of that so it makes more sense. So this is the process we call transcription, where I go from DNA to messenger RNA. I go from the information to the message. So we're going to look at, as this moves through, this process of taking the DNA and the sequence of bases and turning that into a message. So here we have DNA being split apart. So I expose the bases on the inside, and these nucleotides of mRNA come in and base pair with the DNA. That then is split off of my DNA, and now I have a message of the information. This message then gets modified somewhat. They will add ends to it, the front end and the back end. And they'll also take parts out of it. As we mentioned before, they're going to take the introns out, because the introns then don't code for anything. So they're taken out of this message. This message then goes out of the nucleus and into the cytoplasm so it can get turned into the product. So most of your DNA is made up of those introns, those non-coding regions. They do not get expressed. So that means they don't get turned into a protein. So DNA, once again, the sequence of bases, most of which does not get expressed, but we do have parts that do get expressed, and they get turned into a particular protein. So we could think of DNA once again as your genotype, sequence of bases, and then the protein could be your phenotype. So if you have type A blood, that means you're going to produce a protein, this antigen, that's on your cell membrane that gives you type A blood. So that's your phenotype, and that comes from the genotype, the sequence of bases in the DNA. Once again, here we have introns that are not coded. Think about that for a minute in terms of evolution. I'll let you think about that. We'll talk about it later on. Finally, then we come to forensics. We'll come back to forensics. Where can we find DNA at a crime scene? You guys already know the answer to that. Pretty much everywhere. DNA is left behind on anything we touch, but this DNA that we have is known as trace DNA or touch DNA. It's a very, very small amount of DNA. So how do we make use of this small amount of DNA? We're going to show you something that's been done for 3.8 billion years, ever since DNA was first derived in living things, and we're going to take advantage of that thing just for forensics alone. So take a look at this video on replication. So here's our video on replication. This is something that's done in your cells all the time. When you want to make a copy of your cell, you're going to have your DNA make a copy of itself first. So the DNA unzips like we've seen it do before with transcription, but now we have these nucleotides of DNA that come in and base pair. So we're making copies of each one of these strands. And they talk about a leading strand and a lagging strand here. Don't worry too much about that. But the idea is that this is done by nature. Nature has made copies of DNA for 3.8 billion years. So in the end then, I have 2 new copies of my DNA. So we'll do that in lab as well, so that we can make copies of the DNA we find in a crime scene, so we can analyze it and identify who's who in the zoo. So that's a quick review on forensics and DNA. We took a look at this idea of DNA replication. Keep that in mind, because with that information, you now will be able to do a virtual lab called Bacterial Identification PCR. We'll do that next time we meet.